Right. Excuse me. Long time since I've done one of these. Long time since I've talked to any large crowd of people, let's be honest. Thank you all for coming along, and hopefully we should keep you entertained for a little while this evening. There's quite a lot to get through, so I shall breeze through initially without too much pausing and the introductions. Many of you have probably been on these talks before and know exactly who I am. It will be recorded for YouTube, so some of the pages I'm going to specifically put them up on screen, but probably not say much about them. You can look at them at your leisure later down the line. That's who I am. I shan't go into any further detail about that. Hang on. There we go. Right. So as you're aware, we're looking at all sorts of slightly obscure, possibly less often recorded groups than perhaps people normally study. So it'll be just a brief introduction, looking at suggestions of different orders, families of invertebrates that you might not have considered before, maybe some suggested why you might want to look at them, some resources and sort of key characteristics of the groups, and maybe a little bit about how to record and collect some of them. There will be questions at the end, though I'm not an expert really on a lot of these groups, so be gentle with me, and we'll see what we can do. There may be people on this meeting that have more experience with some of the groups than I do, in which case you might find that people can answer your questions. But basically, I'm just going to whiz through to give you a flavour of these things and see if you can maybe be inspired to look at something new. As I say, I'll just look at one or two of these slides briefly so that they're there for future reference, but may not say a great deal about them. As it said on the bump about this talk, there are 32,000 insect species in Britain, which are, a lot of which are relatively well recorded. And the Record Centre does have 66 recording schemes for many terrestrial insect groups, but there are 14 recording schemes for non-insect groups, which is perhaps not that many, and still further some of the things that we're looking at this evening that don't have perhaps a formal Record Centre recording scheme, but there are people out there that are taking records, and a lot of them, the majority of them, you can still use iRecord and things like that for recording them. But really, I just want to raise the profile of some of the things which are as beautiful and fascinating and equally important than any of the beetles and butterflies or hornets that we were just talking about, that perhaps may have escaped the attention of wildlife enthusiasts such as yourselves for one reason or another. So just looking at the supporting actors and the rather than the headline acts on this occasion. I know that there are different levels of experience. Some people are here just out of curiosity, some are quite expert in one field or another, so quite a range of people on the call tonight. I'm not going to read this out just to save time so that we get through things, but it's interesting to read the guidelines for SSSIs with regard to invertebrates, and it just really describes the many of the reasons for looking at invertebrates. I'll just put that on briefly, and then that, and really that the study of them is very important, including these slightly more obscure things. Sorry to whiz past that. You can read that at your leisure, or maybe go and have a look at the reference. But basically, those guidelines highlight some of the problems and some of the motivations to actually study all of the invertebrates where possible. Obviously, it's not something that, you know, most people can perhaps just look at butterflies and some of the more obvious things, and as you're growing up, you learn some of these things. But this is more specialist stuff that we're talking about this evening. But they're all very important, and there is an amazing variety of stuff out there which perhaps escapes attention. with incredible life histories. They're also really important in ecology, but also just from the simple treasure hunt perspective. We were talking about pseudoscorpions before we started, and there are a lot of people out there that really love to find a pseudoscorpion because they've heard that they're so interesting. So there is that treasure hunt element of locating something which is actually perhaps not obvious. So you've actually got to put some effort into it. It's the geocaching of the entomological world, perhaps, and searching things out. There's also, because these things are less well studied, there's a much higher potential for finding new county records, new national records, or even new species to science as well. So that's a great motivation for a lot of people who perhaps are, perhaps you're used to looking at the more common things. So it's quite exciting. It can be exciting looking at these things, but also because of what that report was saying, that there's a lot of these things, there's critical conservation issues, there's wider ecosystem value to these things, and they all have their part to play. So really important. I don't need to justify them any further than that, really. But I'm offering some new niches that people might want to look at, rather than perhaps the ordinary straightforward species that people normally observe. And the warning is that they can become quite addictive. You could become obsessed with it all. But equally, if no one else in your region or your local natural history society are looking at these things, you could become the local expert. It's not a big step from no one knowing anything to you knowing a little bit, to put you in that position. So perhaps you'll be the person that's sort of driving locally the interest in these groups. So we're going off piste. Hopefully the rewards of that will be that we can expand some skills that you have, look at some new specialist areas, maybe make some new scientific breakthroughs, add data to record centres and to any research that's going on, and potentially reinvigorate your enthusiasm for natural history as a whole. Some of these things are really exciting. But there is a but. There are obviously some barriers to overcome in looking at really obscure things. There may be a limitation in the resources that are available to help you identify them. You could find any number of books on butterflies, but you won't find that many books out there on scale insects. Some of the things that I'm talking about, or some areas of if you get into them deeply, may be actually virtually impossible to identify. And there are a few experts to call on for help. They're hard to find. They're very small. In most cases, they're very small, and they are under-recorded for good reason. But don't let me put you off. The other thing is that these days, I'm sure a lot of you have got decent microscopes. Microscopes are much less expensive than used to be, and equipment is much more available. There's all sorts of digital options, which is great. But you could find yourself investing more and more, needing a better resolution microscope and different equipment if you go down this path too far. So it can get complicated. It also explains why in a lot of the cases, for some of these sorts of groups, you find that the experts are professionals working in higher education institutes and other such things, where the equipment's available to them. And I've been fortunate enough over the years to work in some places where I've had access to some fantastic equipment, which has been greatly helpful. But not wanting to put you off, but that said, there are certain restrictions. But as we saw, and again, I'm not going to leave this screen up for long, but over the years, somebody brings out a better book, somebody else brings out 3 volumes on spiders. Suddenly everybody can identify spiders accurately. Some Stubbs and Fogg produced the hoverfly book, and suddenly we're all recording hoverflies. And so with the advent of all of these new publications, and there's some tremendous books out there, the various Wild Guides and Collins Field Guides and Bloomsbury's, and as well as the FSC and RES and LINSOC and FBA doing their more detailed work. There are a lot of things out there that will offer you information. So loads of scope for potentially identifying things that 20 years ago I wouldn't have given this talk, because it would have just been pointless. You wouldn't have had a chance to identify some of these things. But there's a lot more out there. So, you know, some fabulous books on there. The other thing is we have a thing called the internet, apparently. When I first started looking at some of these groups and working at the museum years ago, when the internet started, it was frankly awful. And the identifications, if you knew something and knew anything about anything, you'd find that it was wrong on there. Obviously, that's changed, and the resources on there are incredible. So there's some really great multi-access keys. There are all sorts of keys and very sophisticated recording systems. So you know all of this, the internet, it actually works. But also with Facebook, you actually can now, you can speak to effectively the experts every day. You find something, you can post a picture. All of this stuff is making it so much more accessible to approach some of these things. And of course, talks like this, capturing them on YouTube, you can go back time and time again, and you've got that reference and resource which simply wasn't available a few years ago. So it's all in place for you to sort of stretch yourself and head off onto those new frontiers, which, as I say, you couldn't have done some years ago, but I think we're well placed right now to start looking at some new things. So where to start looking at new things? Being a bit landlocked here, I don't tend to look at marine stuff, and today I'm not going to be casting our net, so to speak, into fresh water either. So this is a rather eclectic and random selection of things, which is possibly based more on what I've taken photographs of and where I've had a bit of experience of finding things, than any kind of coherent logical sequence through anything taxonomic arrangement of things that we might look at. But anyway, I will try and hopefully show you a range of things that are probably things that I found in addition to my main focus. So if you're someone that's sweep netting for certain for hoverflies or something, it's about looking at the other things that are in the net. And so when I turn over logs and stones for millipedes and harvestmen, it's these other things that I've started to look at over the years. So it's looking wider than the normal focus. So to start with, I'll flag up this 8-gap key. Steve Tilling's book is just the classic that will really help. If we're just looking at terrestrial invertebrates, which we are, this is a really good guide to just narrow down what order, what families are we looking at, and it's very good for that. So this is certainly something I've worked with undergraduates on over the years, and this is the thing that opens the doors to the stuff that's not obvious, that they don't just automatically know from childhood. So an excellent book, a mere £8.50, which will open up a whole world of possibilities to you. So I do recommend that. It's not like it's full of colour photographs and that, it's just got some nice line drawings and some decent keys to take you through. So if you've ever used that, that's a good starting point. So roughly, I'm going to point you in the direction of these things, and no particular order, but we will start by talking about the Myriapods. I'm not going to go into any detail whatsoever about them because I've already done talks on them. But before I get on to looking at those specifically, just a quick mention of some of the resources in a sort of general sense. And the Royal Entomological Society keys over the years have been the sort of mainstay for this information on any kind of invertebrate, to be honest. But there are some out of print ones. So the problem is, you know, you haven't got access to all of these things. but there's certainly 4 there that I'll be mentioning later on. And they're old books, you know, from the 50s and 60s, but you know, an excellent way to start. You know, these things taxonomically are still exactly the same as they always were. The anatomy is the same as it always was. So there are some good free resources out there which you can find, and I'll mention those as we come to them again. Obviously, the Linnaean Society as well. There's some good keys out there, some of which are out of print, others which you can still buy at the link at the bottom there. The Harvestman one at the top, the 3rd edition is out of print, but the 2nd edition is available online if you choose to have a look at some Harvestman. And I'm about to, we're reissuing the 2nd edition of the Harvestman Guide with Field Studies Council in the autumn. So enough of that. So I'm mentioning millipedes, centipedes, woodlice and harvestmen simply because that's where my main focus is, that's where my expertise lies, and that's what I'm looking for when I find all of these other things. And for those of you that are perhaps, you might find it a bit daunting, some of the stuff I'm talking about tonight. There's some very tiny little things. These may be obscure enough for you. So these are sort of, there's a lot of resource, there are a lot of expertise out there. There's some very active recording groups for millipedes, centipedes, woodlice and harvestmen. And I would recommend those as a good starting point towards obscure. See, I don't think of these as obscure, some of you might. So that's a good starting point. And there are already webinars with the Tanyptera Trust that I've already delivered all about those in depth. So I'm not going to go into them this evening. But by looking for these things, the interesting thing is, as I get more involved looking for scarce ones of these, by looking in interesting different habitats or where there might be scarce woodlice, the likelihood is that there may also be scarce springtails in the same location as well. And so by looking in one habitat for something, it throws up interesting species in other areas as well. So there's a bit more motivation then, because you may be finding rarer things, shall we say. But anyway, enough of that really, because I've talked for many hours on these subjects before. So those are all the links to previous webinars. So please do go and look at those, they are, in my mind, relatively straightforward, and they're big enough to find, if nothing else. So that's your starting point. By starting with the myriapods, the centipedes and the millipedes, I did in those talks omit discussing the other myriapods, which are the Symphyla and the Pauropods. So I'm just go back one. There are 2 other orders which we rarely talk about because there's not a lot known really. They're not particularly well but closely related, but they do have lots of legs. They have a myriad of pods, many legs, and you can find more information on the British Myriapod and Aspod group website about them. But I'll briefly mention them and show you some reference to them. But lovely little creatures, well worth having a look at and keeping an eye out for. The Symphyla, and I'll show you a picture of them in a moment. Strange little things. They've got 15 plates along the back of the body, so it looks like they've got 15 plates. If it was a millipede, you'd have 2 pairs of legs, or centipede, 1 pair of legs on each of those. But they actually have only 12 segments underneath, with a leg pair on each. And actually the first pair are very small. So when you see a Symphylan, you might think it's only got 11 pairs of legs. So it's got what look like 11 pairs of legs on 12 segments that look like 15 plates along the back. So they're quite intriguing in that respect. The hind end is fairly distinctive, and they've got these little conical cerci sticking out at the end, a little bit like the back end of a woodlouse, actually. And the antennae are very long and made up of possibly up to 60 segments. They don't have any eyes, and they might get up to about 8 millimeters in length. So they're big enough to see, but they're not exactly huge. They are of some economic interest because they can become a pest, attacking the roots of plants. Around 14 species to play with, so not a lot. So it's not a huge number of them. There's not a lot to help you identify them either, unfortunately. But there's a couple of references there, and I'll get back to mentioning Chaos of Delight in a few moments. I'd love to get rid of that word. I don't know if you can see the word you are screen sharing right across the word Pauropod, but I can. It's in my way, but hopefully you can see what's underneath as I move through. So Symphylans, there they are, larger. So as I say, you've got up to 60 segments in these antennae. They're like little diamonds, lots of little diamonds. Quite an interesting antenna on the things. And you can see it's got these 15 segments along the back, apparently. But these are just plates on the back over 12 segments. And actually, if you count them, you'll only see 11 pairs of legs. So quite intriguing. And these little extended cerci at the end. I'm not going to go into any more detail about how to identify these because there's a lot to get through, and I just want to give you a sample that, so if you've seen these things and wondered what they were, it's a Symphylan. There is a full list of the species, and this is the BMIG website, and there's a little bit more information there about where you can find some means of identifying them to an extent. The Pauropods are very small indeed. They only get up to 1.5 millimeters. So I have to admit, I've never seen one. I look often, and I'm in exactly the habitat for them, but I've never found a Pauropod, and lots of people haven't. So just seeing one's exciting. So if you find one of these, well done. Who cares whether you can identify it? It's an exciting thing just to find one. To recognize it when you do, they've got 9 to 11 pairs of legs. They're very like the Symphylans, the first pair are very small. And these appear to have 7 tergites along the back. The distinctive feature really is they've got a little sort of branched antennae at the front. So it's this tiny little creature. It looks like a very tiny myriapod, but it's got these little forked antennae. They're sort of elongate, though one of them we'll see in a moment is a little bit squat and sort of orangey amber. And they have 5 pairs of long sensory hairs that in a close inspection are sticking out at the side of the body. One of the characteristic things that I've been looking for all my life with these is they have a characteristic way of moving. And if you gently blow under a stone, these things may reveal themselves, if they're there, by the way that they move. And they tend to rush a bit and then stop and then maybe reverse and wiggle about and then maybe run a bit and then stop and wriggle about. And it's very characteristic. I've seen video of it, and it is quite strange. So apparently blowing under stones is your next method for invertebrate surveying. And if you find them, well done. I've not found any yet. But there are only 23 species in Britain. Well, I suspect there are more than that. And when people start actually finding them, we may realise that there are more of these things. But there are some references there. Ulf Scheller tends to be the go-to guy for identifying these. But there's some information about Welsh species and various things at those references. But that's what they look like. A couple of pictures here from Stuart Bevan on the BMIG website. So there's these sort of pale typical forms with the branched antenna there, and these more chunky biscuit coloured ones. But bear in mind these things aren't even 2 millimeters long. So, but I look forward to the day I finally see one, and then I can get some photographs of my own. So moving on from things with lots of legs, the myriapods, to things without any legs. And the terrestrial flatworms, their taxonomy is a bit strange. And so what to call a terrestrial flatworm? They're planarians, or the Geoplanidae, or Rhabditophora. So wherever you look, I mean, you might just think of them as platyhelminths, but I'm just going to refer to them as terrestrial flatworms for now. I quite like Geoplanidae. That kind of implies that it's a planarian, a land planarian. But it gets quite complicated if you start searching for these things online or in a book, and quite to know what to call them or what other people are referring to. And there are a lot of flatworms that live in aquatic environments, but these are the ones that have moved on land, and they therefore live in very damp locations, in leaf litter, under stones, under logs, feeding on worms and slugs and other invertebrates. And they actually can get quite big. So in contrast to the tiny little pauropods, some of the flatworms are quite small, but equally when they stretch out, I mean, I probably still could find a New Zealand flatworm in my garden if I searched. I've been battling them for years, but they get up to sort of 15 centimeters long when at full stretch, and then shrink down. So they're pretty big, sort of 6 inches in old money, large beasts, and they stretch and contract. So you never quite know. If you wet them, and put them in a wet container, you might see them stretch to full length and appreciate their full size. They're incredible things. They're seemingly invincible. You can cut them in half, and they'll regenerate from that. You can cut them down the middle, apparently, and they will still regenerate. There's a species, one of the species in Britain recently found, and it virtually fragments if you handle it. If you upset it too much, it fragments into small pieces, and they can regenerate into new individuals. So quite a difficult thing to kill off, really. But they are very susceptible to desiccation. If I want to kill off New Zealand flatworms, you know, I'll kill them, but I'll leave the bits in the sunshine to dry out, just in case. But the first time I ever saw one of these at the museum, someone brought one in, the first record for Sheffield of a New Zealand flatworm, left it in a jar overnight, and the conditions presumably didn't suit it. And under the stress of that situation, it dissolved itself. So their mode of feeding is often to sort of dissolve their prey. But they will autolyze themselves as well in captivity if you're not careful. I've kept them perfectly well without that happening. But yeah, under stress, or if the conditions are too cramped, they just sort of dissolve themselves. It's quite an unpleasant experience to see. So intriguing beasts. They have an extendable tube for feeding halfway down on the underside. But that also is a feeding apparatus. It's also the anus. So they have to have completely digested their meal before they can feed again, because it's the same place. So they digest it, excrete before they can start again. An interesting concept. The native species, we have a number of native species and a lot of alien exotic species which have been introduced. The native species are identified relatively easily as having 2 eyes, whereas the introduced species have lots of eyes down the side. So this is one of the native species, probably one of the commoner ones, Microplana terrestris. You can see down here, that's little eyes, 2 little beady eyes at the front. They're sort of hardly eyes, but they're light sensitive structures of one sort or another. It's a fairly featureless creature. It's sort of a blob. It has sort of creases and sort of Not really, doesn't look segmented, but it does sort of have these sort of occasional creased marks along it. I'm not going to teach you how to identify every native flatworm that we've got, but that's the all grey brown Microplana terrestris, whereas its cousin Microplana scharfi, quite different, bright white, yellowy, white, cream, whatever. And you can see the eyes there, and they get long and they get short, and this one's not, it's not like it's a snake with a meal in its gut. I think it's partially contracting. Also, it could be that it's got eggs which it's about to release potentially. I'm not sure exactly the detail that's going on there, but an obvious looking beast, 2 eyes. When we start moving to more the exotic introduced species, you can see lots of eyes, lots of these little dots down the side of this Contychia ventrolineata. I've got a lot of these in my back garden, and it's got 2 pale stripes down the back, or 1 dark stripe if that's the way you look at it. And on the underside, you can see these dark bands as well, which will help you to identify. And if you look at it more closely, it's got these beautiful blue flecks. This pattern along the back is actually sort of almost like it's metallic paintwork, but to all intents and purposes, when you see them, they're just little black worms. But in detail, they're quite attractive. And you can see the underside with these stripes, and again the eyes there. So that's Contychia ventrolineata. There's another Contychia there, ventrolineata, next to the notorious New Zealand flatworm, these days named after Arthur Dendy, Arthrodendius triangulatus. And it's quite a big thing. It's quite obvious. People often wonder what these look like. Sort of a chocolate brown with pale edges and flecked, speckled edges. And yeah, it's a big old beast. But when you see them curled up, when you often find them, they're quite attractive and these sort of rosette patterns. And there's a lot of mucus around. And if their eggs are there, their eggs are very distinctive, like little blackberries, and they are quite large. I mean, they're pretty much the size, sorry, blackcurrant, not blackberry. They are pretty much the size of a blackcurrant. And they're quite distinctive. So if you see these things in your garden and wonder what they are, could well be the New Zealand flatworm, and that's really, I believe, still a notifiable pest which should be destroyed, because it's believed to do no good whatsoever to our native earthworm populations, because that's what it feeds on. Something like Contychia ventrolineata, it may be feeding on earthworms, it may be feeding on slugs. I'm not certain, but I believe that certainly some of them feed on slugs, so you might consider those as a beneficial thing if you're a gardener. But they're all introduced, so the ecosystem balance is being shaken somewhat, so none of them are really great things to encourage, I imagine. Then they're beautiful things like this. So a lot of these are really attractive flatworms. So this is Australoplana sanguinea, and it's the one that tends to be called the Australian flatworm, but there are quite a few that are potentially Antipodean at the very least, and others which you could consider are an Australian flatworm, but this is the Australian flatworm, at least in this country it is. Beautiful orange fading to yellow in this particular example. And I think some others are just bright orange. You can see the eyes all dotted along the edge, so lots and lots of eyes rather than these 2 at the front. This is another introduced one, Australopacifica atrata. Australopacifica is a sort of catch-all generic name for some of these potentially sort of, they're from Australia or South Pacific, or they're basically overseas species. And this one looks a little bit like the first one, Microplana, but it's got a sort of paler edge, It's similar to the Conticia. It's got like one dark band on the underside, and but lots of eyes, perhaps not clearly seen in this photograph, but more than the two at the front end. So you know it's not one of our native species. I think this was in Anglesey, actually. But that's all. I just want to show you some examples. There's some really pretty, really attractive looking flatworms. I say attractive, they're a pretty horrible beast when you say they're very slimy and unpleasant in that respect, but quite pretty things, with some of them with like hammerhead extensions at the front and all sorts. The best sources of reference are, there's the NNSS produce ID sheets. Bug Life have got ID sheets. There's a good reference if you, and it's available online and various places that you can find it. There's one there. Hugh Jones some time ago wrote for British Wildlife a guide to British land flatworms. And, so that's your best resource really for that. He has his work, certainly I've seen an updated version of that with some of the new species which have been found subsequently, including this beast down at the bottom there, which was recently found in Sheffield, and there's a paper there about that. Sceneplana variegator. So these things are cropping up all the time. Often garden centres might be moving them around, the eggs are in soil. So keep an eye out for them. Hugh Jones is very helpful, and he no doubt would want your records for anything you can identify, and will no doubt help you to identify, has helped me to identify species in the past. So that's the flatworm email address. So that was your flatworms. Here we go. It's a mad race through things. Back to legs though, things with legs. The Bristletails, Firebrats and Silverfish, the Thysanura, as they used to be called, and Diplura. Thysanura really are now Zygentoma. Between the two of these, they're grouped together for a variety of reasons, but from my point of view, it's because they are in the Royal Entomological Society guide handbook from 1954, which is available online as an out of print. I've put an addendum there. There's one particular error on there if you do go to use that. But you know, yeah, it's 1954, so knowledge has moved on, but it's still a good source of a starting point for you. With these beasts, one of them has 2 spines at the back and one has 3. The abdomen with 3 projections in Thysanura or Zygentoma. And they have very long, distinct palps at the front, which are longer than their legs, and they're reasonable size. They're about a centimetre in size, so noticeable. You'll have seen them at the seaside and limestone dales and various places. For absolute identification, for a lot of them, you could really do with a male specimen, but there are only about 9 species to choose from. The Diplura have just these 2 cerci at the end with no central filament, and their mouth parts are sunken inside. You can't see the mouth parts, and they're much smaller, about 5 millimeters, and there are only about 11 British species. If you look online, you'll find that there are sometimes people talk about Bristletails and get them mixed up with Springtails, and we'll call Bristletails Collembola. When I was researching this talk, I was just finding the Bristletails called Collembola, which is nonsense. But they can jump, so people think that a Bristletail, they find it's a jumping thing, so they think it's a springtail, and they're not. So be warned if you're looking online for them. And they're no longer considered to be an insect, but they still belong within the wider Hexapoda. So these are your Zygentoma that used to be Thysanura, and as I say, there's 3 spikes at the back, really, in a silverfish. They can live quite a long time. Silverfish, they're unlike insects in that they will molt, molt again and molt again, and sometimes they're sexually mature through that process. So as they molt and grow and keep going, they can last for quite a while. One of the interesting things is if you get fed up with seeing silverfish at night when you turn on the light in your house, they may save the planet yet. Apparently there's a chemical process that they use for breaking down wood that they feed on, or cardboard and paper and the like. And it's like a bioreactor within the body. And people are doing research at the moment to see that there's a potential that there's something about this that might be used for creating biofuels. So who knew silverfish are going to reverse climate change or something? I may be reading too much into that. So there we are, silverfish. Their eyes are quite wide apart in the Thysanura and the Zygentoma. It's quite a flattened body, and certainly in the silverfish, you can see they're very silvery, shiny, scaly. And the Lepisma saccharina, you'll also find that there's firebrat. But there's since 2016, there's another sort of hairy version of silverfish. So if you find a particularly sort of scaly, hairy version of a silverfish, it could be this new species of Lepisma longicordata, which is not well recorded anywhere much, but it's around. So there's a new one. So silverfish isn't just a silverfish anymore. There are other options. The Bristletails, these are the sort of the Machelidae, which are, their eyes are closer together at the front, so the big compound eyes, usually sort of touching in the middle, like they're wearing sunglasses. More often than not, there'll be a Delta species, and there are a few others. And then to separate them, it's usually a matter of where their additional ocelli relate to their eyes, so whether they've got extra ocelli underneath or in these back corners or all the way underneath, things like that. But the guide that I showed you earlier describes that. So you get quite a good head start, even though it's quite an old book. So do record the Delta and related species. There's one Delta, I think, which is recently discovered new to science, which is in Wales. So they're out there to be found, pushing back the frontiers. So more two-tailed things. So these are the two-tailed things rather than the three-tailed things. Diplura, I won't go again into too much detail. The book covers things. The way to describe to identify them is looking at sort of microceti on the tergite. So you need a decent microscope. But I see these all the time. I used to think these were the Symphyla, but Symphyla don't have much of a tail at the end. These have got the long tails. Interestingly, pretty much any appendage on a Dipluran will regenerate if it loses it. But there are only about 11 species, and I don't think anybody's really recording these. I suspect that not many of you have recorded a Dipluran. So there's a thing you could try, and knowing that there's a decent free copy of the book out there to have a look at, it might be something you want to have a look at. I mentioned briefly this earlier, and I'll give it a better plug now. In terms, I've not got great pictures of Diplurans. I would heartily recommend a website to you if anyone hasn't looked at A Chaos of Delight, which is Andy Murray's website, not that Andy Murray, the Andy Murray that knows how to use a camera very well. And most of the things I'm talking about this evening, he's got the most immaculate, fantastic photographs of them from various parts of the world, and including a lot of British species, a lot of really useful information. So big up that website. You really should go and have a look at Chaos of Delight. And throughout, as I'm talking, you'll see a reference to it cropping up, because it really is a great place to see stunning images of some of these things. So it's a good place to go and have a look and see what Diplura might look like beyond the pictures I've shown you. So give them a go. You go on Chaos of Delight, you'll find some Pseudoscorpions or false scorpions. 29 British species, I believe, at the moment. They're an arachnid. Very, very popular in that once you've seen one, you're smitten because they're a delightful beast. They tend to run backwards, and then they might have a go at you. You know, they'll wave their claws to say keep away, but they'll run in reverse as well as they do as they do forwards. Basically, it looks like a scorpion without a tail, but they're very small. They don't get bigger than 4 millimeters, as we were discussing at the beginning with someone. And I never thought, I have to say, they can be tricky to identify, or at least I thought so. But the Field Studies Council Aid Gap Fold Out Guide has proved that they can be accessible to everyone, and that's excellent. We road tested that and it works really well. And they've got, as you can see, I mean, they're fairly obvious. They've got these claws at the front which just look like scorpions. They have a pair of smaller jaws in between that. They have 8 legs. They have maybe no eyes or 2 or 4 eyes. You'll find them under bark and stones and leaf litter and in moss, with a lot of other invertebrates. But also you might find them hitchhiking on flies or harvestmen. And I haven't got any photographs of that, but they are just literally using those claws to hitch a ride on a fly and dispersing perhaps from one dung heap to another, one compost heap to another, by flying on a dung fly or whatever. So keep an eye out for those, but they're tremendous beast. So that's Gerald Legge and Francis Farcox's guide, which I heartily recommend. Gerald Legge also has an excellent website with a lot of information. This is a screenshot of that, so you can find more information about each of the species, to dig a bit more deeply into if you're trying to identify them. Liam Andrews also recently, he runs the Facebook group and recently did a talk for the British Arachnological Society at that YouTube address there, so you can go look at that. So there's like a 15-minute introduction which will give you an update on their distributions and new species that have been found or refound around Britain. And hot on the tail of when you write one of these things, it's virtually with the intention that it's out of date as soon as possible, so that people can't find it in here and realize that it's a new species, which is exactly what happened with the Harvestman Guide. And yes, lo and behold, there's now a new Neobysium simile has been found, new to Britain, 2017. So there's scope for new discoveries. I'm going to have a quick look through the Pseudoscorpions in a moment. That's the Facebook group. Do go on there. A lot of useful information, a lot of enthusiastic people getting very excited at finding their first Pseudoscorpion and then getting hooked to look for more. The world of Pseudoscorpions there. This key from the Czech Republic and Slovakia is really good, in that it's more of a dichotomous key with a lot more information. So if you're trying to get to grips with Pseudoscorpions, the Linnaean key is out of print, so that's very hard to get hold of. But I haven't checked exactly how much of an overlap there is, but it looks like most of the British species are in that Czech key as well. So do use that as a good resource for more on Pseudoscorpions. So Pseudoscorpions, I mentioned they've got a big claw and a jaw. And the two commonest you probably find will be the Neobysium carcinoides and Chthonius ischniceles. Difference being that in Neobysium, the hand of the jaw here is narrower and somewhat smaller here than the claw hand. So jaw smaller than claw. And in the Chthonius side of things, the jaw is almost, the hand of the jaw here is more like the size of the claw hand. So a small jaw, you know what I'm saying. The way you often look at it in the key is these large, very large jaws here are virtually the size of the cephalothorax here, whereas as we'll see in a moment, in the Abyssium, they're much smaller. While we're on looking at some characteristics, another that used to be called Chthonius, it's now Chthonius tetracalatus, has this little dimple on the claw here. So you're following it through the key and you're looking for a little thing like that. So there's subtleties about them, but once you know what you're looking for, they're not that difficult. So this is Neobysium carcinoides. Probably, if you're going to find one, it might well be this. It's got, you might be looking for the pair of eyes, a couple of eyes, various characteristics as you work through the key, but just highlighting there they are on that. But tremendous beasts. You can see waving their claws at me fairly aggressively while I was photographing it. But beautiful things, covered in a variety of hairs, which can be quite useful in various points to help identify them. If you get close up to the to these claw hands here, you can see an arrangement of spikes there, and on Neobysium carcinoides, you've got this big claw, little claw, little claw, little claw, big claw, small, big, small, big. So you've got this arrangement of little claws with big claws along. A subtle detail that requires good backlighting to see, but it's not like it's the only thing that's required for identifying it, but it does help you to separate it from something slightly rarer. So size and shape of teeth is useful. Maybe there might be a number of setae on the back of the cephalothorax. That might be a helpful feature that you, something which will crop up in the key. You can see here from this picture, this is where these jaw hands are virtually the size, give or take. I can't do it on the screen. I'm doing that on the screen, but my cursor there, you can see that that's very large. It's virtually the size of the cephalothorax. Whereas on this, going back to the Neobysium, it's much smaller than the length of that cephalothorax. So when you come into the key and you're wondering what that means, it's just these are smaller than the length of that, and they are smaller than the width of these pincers. Okay, so there you go. So these big jaws, about the same size as that, big jaw, about the same size as the pincer, the hand of the pincer. So things like that, pretty helpful. This is particularly helpful. Chthonius tenuis, it's got boxing gloves, it's got nice black signaling hands. It's a tiny little thing, but you can see these little black hands waving at you threateningly as you find them. So that's quite a nice little feature. This is Bronchus lubricus. Might well find this in your compost heap. One of the characters you're looking for here, it seems a bit obscure, but you can see it with the right lighting. There's this regalia and the end of the jaw. It's actually used for making silk. So they will make little silk chambers, and that's where they produce this silk. And the height of that bump can be a useful character to separate it from something else. Something like, and the other character, go back to here, on the back of the abdomen of one of these species, you can see it's a single plate right across the back. So nice straight plate right across the back. When we get to these chinetid type species, it's split down the middle. So you've got these sort of dark plates either side broken with a central line. So that's a very obvious, that immediately splits things. So you go through the key and it very quickly splits them into two, so that's a useful thing. So they split in two, rather like the tergites are on the back. There may be setae of one shape or another, so these have got clubbed setae on the back here, which is a helpful feature. There may or may not be long sensory hairs called Trichobothria on the claws or sticking out of the back. This one doesn't have them. There may be that some species do have these long sensory hairs at the back. So those are the sorts of things that you'd be looking for. And we were talking earlier about these sensory hairs on the legs as well, to separate some of these dung and compost species. There's this character on the back leg. There's this sensory hair on the fourth tarsus, which you can see it. It's not outrageously difficult to see, and you just have to work out whether it's in the middle or whether it's towards one end of the tarsus. So need to have a decent hand lens at the very least to see that, but you can see it with a hand lens. So Lamprichernes sauvigny is something which I found regularly, not often, but regularly in compost heaps, and that sensory hair might be what you're looking for. If it's in a different location on the leg, then it might be Lamprichernes chisori or Lamprichernes nodosus. These are notoriously tricky to separate, but there are ways and means. You're looking for the shape of this bulge here. So they both look very, very similar in all respects. That and that look very similar, the one on the left and the one on the right. But this bulge here and this bulge here are slightly different. Now that might be difficult to see, but it's a reason to join things like the Facebook groups. And this is where you've got this huge advantage. When I started looking at Pseudoscorpions, I puzzled over this for years, not really knowing quite which was which, despite the best illustrations. But you go on Facebook and you've got Gerald Legg and others. This was just taken literally from the Facebook group some time ago, and people are trying to help you identify these things, helping you to separate them so that we can record them accurately. So do join, these Facebook groups for all sorts of the soil biodiversity group covers a lot of the groups I'm talking about. Very helpful people out there that will give you this level of information to try and tweak your skills to recognize your difference between these closely associated species. Just finish off with it, there's just a bunch there. This one here, there's a circular depression, Caridium museorum, which will help you to identify that one. This thing has got what's called a point. Some of them have a little epistome at the front of the cephalothorax, it's quite sharply pointed in Carpenteri. So there's a host of different things, but they do all look the same. And when I started out on these, it was very hard to do Pseudoscorpions. It's considerably easier and much more accessible now to do so. To an extent, that's true of the mites and ticks too. So the acari, let's have a look at these. I'm not going to go into these in a huge amount of detail. I know I'm no expert on these. Normally, acari are broken down into perhaps 3 groups, 4 groups, the Mesostigmata, the Oribatids, and the Prostigmata. Plus the Exodida, which are the ticks. We'll include the Endiostigmata too, though they're slightly smaller and more difficult to get your head around. So a bunch of different things, and we'll look at those a little bit now, but not at any length. There are over 2,000 species, so plenty of scope. About 1,000 of those living in soil. So you can't fail to find them. They're hugely abundant. There are masses of mites and to an extent ticks, but mostly mites. The Mesostigmata mites belong to the parasitiforms, and so do the Exodida. But just look at the Mesostigmata. There's about 450 species, lots and lots of them. They're all predators. They're recognized by having a spiracle or a stigma, like a hole in the side. in the middle of the body, along at the end of their breathing groove, which is called a peritreme. So mesostigmata means basically it's got a stigma in the middle. A varroa mite that you may have heard of that cause problems with honeybee hives, that belongs to that group. I'm not going to say a lot more about these things, but I will recommend some excellent references in a moment. The Exodidae, the ticks, these have got a very barbed spike at the front for mouth parts for piercing and anchoring itself into its host, and they just suck out the juices, the body fluids, usually blood. In terms of the difference between these, they don't have this groove along the side, this peritreme, this breathing groove, but they do have the stigmata, but it's slightly further along the body. But that's sort of how we distinguish ticks from the predatory mesostigmata. So those are some ticks. So this is Exodius ricinus, and the reference to these, I mean, this is you expect them to be bloated up, big fat females that are feeding on blood. I don't have a picture of such a thing. And this is found around where there are deer and sheep. We don't get many ticks, fortunately, around our way, but they are increasing in number and dispersing far and wide. Paul Hilliard's book, The Ticks of Northwest Europe, is very good, but out of print, and I don't think it's available online anywhere at the moment. But if you can get hold of a copy of that, that will help you to identify ticks. I used to use it to identify things being pulled off the heads of children at the Children's Hospital in Sheffield when they brought them across to the museum and said, "Can you tell me what this is, please, that we've just pulled out of a child's ear?" Happy days. But I no longer have that book at my disposal. The other mites, the one that's perhaps reasonably well known is the Aurobated. The Aurobated largely decomposers, about 670 British species. They're like a crash helmet on legs, like Darth Vader helmets on legs, if you like. Just a smooth blob of hard nothing, really. They're like a little beetle mite. The mouth parts are hidden under a hood at the front, and they have one of these. I mentioned the name Trichobothrium earlier. It's like a hole with a seta sticking out the side on each side of those, and some of them are these Damiid mites. You see them, they're like little spiders, or they cover themselves in soil, so normally it looks just like a little bit of moving dirt, but they're pretty common, and yeah, so that's a nymph of a Damiid that's trying to disguise itself. There are other within that, the Aurobateds, there are 300 of these astigmatina, which include the dust mites, and these are fairly pale. They're not armored in the same way as these, and they just have a few sclerotized sort of hard bands on the underside, and the mouth parts are actually visible from above, and they don't have these Trichobothria. So that's about all I know about mites that I can share with you, but I'll show you some pretty pictures. There are, in addition to those, the prostigmata. These are ones that you might well be familiar with, if anything. About 900 species, so again, scope. All sorts of different lifestyles, life histories, including a lot of plant galls, and there are aquatic ones, but also the spider mites and the velvet mites, which you might be more familiar with, and the sort of the larval mites that you see attached to insects, and just cemented and stuck in as they mature, before they drop off and become adult. They're usually fairly soft, often red, tend to have broad shoulders. In certainly these are the sort of velvet mite types, which are very obviously got these quite broad shoulders, and they're sausage-like legs. Is the way it's been described, and they've got these. Their legs will seem to be similar size, like a string of sausages, really. And there are various other characteristics. You see there's a gap between the two at the front and 2 pairs of legs at the back as well, and 1 or 2 other things to do with the details of their legs and claws on there, and have some fascinating thumb. Well, the end of their tarsus is bent down before the claw, but I shan't worry about that. It's not something we're going to need to do. I'm not going to mention more about the Endiostigmata. There's about 12 species of those. They're very, very small. They may have Trichobothria and they have eyes, but I have no pictures and I can't say that I've ever seen one. But I'm not dismissing mites and ticks. They're very, very interesting indeed. Red velvet mites, something which you might commonly see. Trombidium, Trombidium rostratus. They're quite attractive Erythraeus species. I mean, this one's particular beauty. But you see those trotting around in soils, and often probably a lot of people might see them and wonder what on earth they are. So they are these velvet mites. Now, the best place to go is, there are 2 excellent videos that you can find. There's a collecting mesofauna video which Matthew Shepherd has produced about how to collect these things. But on YouTube, everything you wish to know about mites, on that is Matthew Shepherd doing what I'm doing now, but identifying mites. And that's excellent. That's everything you could wish to know. The Soil Biodiversity Facebook group is also a way of connecting with other people that recognize some of these things and other things, such as springtails. And you get Matthew on there, various other people who will help you identify some of your tricky things. But more importantly, if you're in the Facebook group, if you go to the files section, you can find a copy of a key, which is a fantastic key. And Matthew Shepherd and Felicity Crotty have written this key. It's full of fantastic photographs of all the characters to identify these things down to families. And it's mind-blowing, really. It's really detailed and helps you to from recognizing all the anatomy and characteristic features and working through the key. And it's still a test version, and it's always a work in progress, and I expect one day it will be published at some kind of endpoint. But for now, it's very generously available to those of us who would like to identify our things to families. And there are records of families. There are so few records of these things. If you go on iRecord, you can record things as a family. You don't need to know what species it is. Matthew's happy to accept family records as an encouragement to people as much as anything else, I guess, but also it shows that family was present at these locations, which is more information than is probably available at the present time. So do have a go with mites and ticks, they're interesting. Additionally to that, there's the recording scheme website there, and an email address if you have any specific questions. There's a soil biodiversity website there with photographs and more information. Bristol University have got a tick identification key. Chaos of Delight again has some lovely pictures of mites. And our very own Northwest Invertebrates, if you go onto the website, there is actually a publication about mites. Someone has been recording mites in the area around the Northwest, and maybe you're on the call now, who knows. But so you won't be the first person in the Tanyptera Trust area to record mites, but there is information on there if you live there in this region and want to have a look at more and see what's been recorded. Springtails again, here we go, whizzing through, but not going to mention them in great detail because I'm not an expert. There are people out there with much more knowledge about springtails than me, but they are really a great group, growing in popularity, particularly since Steve Hopkin produced that just mammoth book on springtails. If you've never seen it, it's just awesome. Full of photographs at the end and of every little detail, but an amazing book, and that means that more people are doing springtails these days. In addition to that, Chaos of Delight, again, specialist in Andy Murray in taking photographs of springtails, as is Jan van Duinen, as is Eddie the Bugman. I recommend, if you're interested remotely in springtails, those 3 sites in particular are awesome. Eddie the Bugman's Flickr site has some tremendous photographs, and I say Jan van Duinen's got some incredible pictures of what are very, very small things. So all 3 of those, hats off to those guys for their photographic skills. Colembala.org, loads of information on there. There's stuff about nature on Nature Spot, and there's also Colembala blog spot for some identification things. But a quick look at some of them. Colembala basically means glue rod, and they have I think people tend to think it's the springing thing at the end of the tail that gives them their name, but it isn't. The thing that's unique to them is they've got like they can exude a tube out of the sort of chest area, I guess, top of the abdomen, which is used for osmoregulation. It's helped sometimes to help them self-right when they've fallen over, and also it's sticky, and it will help them to grip slippery surfaces. So that is the key character that makes them a Colembalan. But they do have this furca at the tail end. It's a springing organ. Something like the small grub-like ones, it's rudimentary, but some of the more insect-like ones have got this spring, which gives them the obvious name that they do go ping as this thing catapults them into space. There are around 250 British species, so it's a good group to tackle. There's plenty of them. There are the Poduromorphs, which are the sort of small grub-like things, which have this just a reduced furca, so they don't have much of a springing organ. The Entomobryomorphs, as the name kind of suggests, are the more insecty ones, and they have distinct appendages and a distinct springing organ. And then there's the Symphyla and to an extent the Neelepleona, which are these globular species, which again have quite a well-developed springing organ. I'll take a quick look at some of them. This is one you can learn to identify quite easily. Orchicella cincta is one of these entomobryomorphs. It looks like a bit like an insect, as it should. It's got a dark band down the back with a sort of yellow band in front of that. It's got this pale line at the base of the antenna. It's very hairy, and you can see that very clearly. It's sitting in leaf litter or whatever, and wherever you can spot these things with this dark band, this yellow band, these pale antennae, and it's let's just get back. It is a pretty straightforward springtail, so it's encouraging to think that there are some out there that are quite straightforward, and I'm probably still at the stage of knowing the straightforward ones rather than some of the really difficult ones. You can see here this is Orchicella. It's quite large as well. And there's a baby Orchicella, but that's also more indicative of the size of perhaps some other springtails as well. So hairy and banded. This is Orchicella villosa, so very closely related and very hairy beast, and that's relatively straightforward to recognize. I'm not going to go into all the details today, but then there are some of these more grub-like ones, and these are actually these are a treat to find. These are very nice, very easy to recognize in many respects. This one's a sort of mucky grey and has 3 ocelli, so it's got a pair there and one solitary one there. Neonura muscorum, find that fairly often in dead wood under bark. And then this is Monobella grassii, which not well recorded. It's but easily seen when it's there. If you recognise it as Monobella. It's got 2 eyes, so one at the front, one there, one there. but it's this lovely sort of orange colour and relatively easily identified. So look out for that. You may be, it might be a new county record for some people. I think it's fairly widespread and widely recorded now. But I think under-recorded for how relatively easy it is to identify. This, on the other hand, really distinctive creature. I only realised I'd seen this when I started doing the research for this talk and found it tucked away in the corner of a photograph of a woodlouse. And then looked and found I'd taken more photographs of it. But this is Balabella brunneri, and this was new to Britain in just a few years ago, I think perhaps 4 years ago, something like that, and is only known from Shropshire that I'm aware of, and this is from South Wales. So this was new to Wales. When I didn't realise I'd found it, but I found it in South Wales, new to the country. But it's bright pink. It's very distinctive. It's got just a couple of eyes. The colembla.org is fabulous. It's got a lot of colembla of the world, and this is distinctive enough to recognise it from there. But also Pete Boardman, I think, originally found it a few years ago and wrote a paper about it. So new species, look out for it. Be the first in your county to find this really distinctive beast. So there's your target. The globbies, as they tend to be called, globular Symphypleona, Dicertomina saunders is probably one of the most recognized or most popular. It's a very pretty marble, multicolored creature. And but there are others like Dicertomina fusca, which bright red in that case, or darker purple. All sorts of globular springtails with great patterns, and they do ping quite quickly. They do tend to spring away, and if you drop them in alcohol, a lot of springtails just float, and quite hard to preserve a springtail, but with perseverance they do sink with a bit of detergent. But often you do have to collect most of the things I've talked about this evening and preserve them to look under a microscope. But springtails, globbies, brilliant creatures. In fact, in Sheffield, in the last few years, new species to science have been found, and not by me, but they can be found anywhere. And this is a Lacma fusca, which is a springtail, sort of mucky brown thing. I was looking for bark lice on tree bark and saw these, and it's quite big. I thought it was bark lice, but a wingless bark louse, but it's a springtail and relatively straightforward to recognise. And then this little fella, which is used to be called Tomocerus longicornis. Blow on this and it will curl up its antennae, or the antennae curl right up. It's got very long antennae, as the name suggests, longicornis. Various states of undress. The scales rub off on a lot of the springtails, and from virtually covered in silver scales to virtually nude down there. But this long antennal segment will curl right up, and that's very handy for helping you to identify it. Another link there to another useful place for identifying the Tomocerinae. So we're moving on, and time's running out. I'm talking too much. So thrips. What can I say about thrips? They're fabulous. Thrips are great. Thysanoptera, fringe wing, that means. And as you can see, they have wings which are largely made up of like a lot of hairs. A thrips, if you learn nothing else tonight, this is what you will learn. It is a thrips, and they are thrips. It is as in sheep, many sheep, and one sheep, many sheep. One thrips, many thrips. A better analogy, I think, for you to remember might be a Greg's. You don't go into a Greg, you can only go into a Greg's. So you can go into several Gregs, or you can go to a single Greg's. Well, there are thrips, and there is a single thrips. So it's very tempting to say you found a thrip, There is no such thing as a thrip. There they are. They're probably known as thunderflies by a lot of people, little tiny flies which are attracted to bright yellow. If you're wearing yellow perhaps in the summer and get covered in little black flies, it may well be thrips. Diagnostic characteristics, these feathery wings, they have these stompy little legs. It looks like there's a bit missing. It looks like there's no claw or anything, but they do have like a bladder at the end that helps them to adhere to surfaces. They also got bizarre mouth parts. They've got these piercing, pointed mouth cone, as it's known, on the mouth. And they're all left-handed, I think, and I think they all are. They lose half of their mandibles during development, so they're asymmetrical with their mouth parts, and they only have a mandible on the left-hand side, but they have this sort of beaky mouth cone. They do look all very, very similar, but there are about 160 species. Fantastically interesting. I mean, the males fight. You can watch them doing all sorts of battles, and they produce pheromones, alarm pheromones, like Hymenoptera. They're fairly easy to find, but very underrecorded. Probably more because they're quite difficult to mount and identify. You really need to make a mount on a microscope slide. I'm currently experimenting with using glass Loctite as a mounting medium. I'm trying a few thrips on that to see how that goes, because it's quite difficult to get hold of mounting media when you're not employed by a university, which I am not currently. So they're quite varied when you look down at the detail, but equally they look very similar initially. They're fantastically intriguing in that they remind me of Hymenoptera in that they have haplodiploid reproduction, which means that fertilized eggs produce females and unfertilized ones only produce males. So they have castes. Well, they get some have soldier castes like ants do. So producing pheromones and having this reproduction and having sort of different forms of adults, a little bit hymenopteran in some respects. They tend to hide away. You've got to look hard for them sometimes. They might just be on the surface feeding on pollen, as these are, but they might be feeding on dead wood or leaf litter and bracket fungi. You might find them in a number of places, but often they're hiding away. So they might be deep inside the flower head, deep inside here, or you see there, that's inside ragwort, an individual flower ragwort, and inside there is your thrips, yeah. And several thrips inside these seed heads, and some grasses that are sorting through, which you could easily miss. So you've got to look hard for thrips sometimes. They can be very plant specific, so that's a really helpful aid to direct you for your identifications. So general sweep netting for thrips isn't necessarily that helpful, because you probably swept through 10 different plants before you find things. So you can lose a lot of information by sweeping for thrips. So it's better to focus your attention on privet today and ragwort or whatever, and then that will give you a clue sometimes towards their identification. I'm not going to go into much of this detail. I'll put it up so you can look at it later. Some are crop pests, so they're significant animals, but equally some of them may pollinate, but then again they also feed on pollen, so maybe they're causing damage, but also some of them are predatory on other pests. So swings and roundabouts as to whether they're a good or bad thing, but they certainly have a number of different lifestyles. There are only 3 families in Britain: Thripidae, the Aleothripidae and the Phleothripidae, often feeding on flowers, but sometimes on leaves, sometimes fungi, as I say. So just looking at some thrips, I photographed these specifically for tonight. but I haven't had a chance to mount them and identify them correctly as such. These were on oxeye daisies, so so maybe oxeye daisy thrips. There is a Haplothrips leucanthemi, which is specifically likes oxeye daisy. The larvae of which is bright red, so that's a helpful thing to find. But again, it's helpful to know what you've looked on. So what I'm going to show you are plants with some thrips. So these are the thrips I found on hedge bindweed, and there are some, according to the Kirk book, which I'll show in a moment. There's a list in there of plant species and the thrips that are associated with them, which is helpful. So potentially Thrips major, Thrips fuscipenis, possibly something else, which will be revealed hopefully when I've mounted them. And this was specifically on privet, and there are species which favour privet over other things. So you get the point. You can there are a number of species of plants, some of which are very specific to thrips. So investigate a specific plant potentially if you're looking for thrips, and that will narrow things down. This was on ragwort. There's ragwort Seneccio jacobea, and there is a Haplothrips seneccionis, which this may be. You can see it's deep inside the ragwort there, hiding away, quite difficult to find. But they're not disappearing. They don't fly off as soon as you find them. They tuck themselves deep down inside, so quite easy to dig out of thrips. Likewise, meadow buttercup could be potentially, this could be Thrips flavus, this yellow one down here. This may be Frankliniella, not sure, certainly not from that photograph. But interesting things not to be overlooked, and there is help at hand to help identify them. There's a fantastic website with a lucid key by Collins, Hastings and Mound, and I think it might be Don Collins that wrote it, not sure. The Thysanoptera Britannica hibernica, British thrips, and that will guide you through and also show lots and lots of electron micrograph type, or at least high resolution images of the various parts that you might be looking for. So do check that out, that's really excellent. But also the original Royal Ensoc key to thrips is available online. So that's excellent. So you can download that and have that to hand, as well as having this sort of more modern support of the Collins, Hastings and Mound. And as I've just waved around, there's William Kirk's book, which you may be able to find a copy of, which is great. It's got colour pictures and keys and various things in there. But anyway, if you wish to do thrips, do so. Right, coming into land, earwigs. There are only 4 native and 3 alien species. Do look at earwigs, they're overlooked. They have the pincers at the back, you know, an earwig, I'm sure. They are they have very small wing cases where they fold away their fan-like wings, and only really the lesser earwig, which is this one here, tends to readily fly. The rest of them tend not to fly very often. Omnivorous, feed on all sorts of things. You may have found females with a little nest of baby earwigs, which are usually the common earwig, which we'll look at in a second. More information on the Orthoptera page. The original RES key to Dermaptera is available online at that link, though something more like the Harley book by Marshall and Hayes will probably give you a bit more information than that. But not much has changed in the world of earwigs in the last, well, since 1949. But there are 1 or 2 species that are probably less common than they were in 1949, if anything. So just looking at 3 of them there: Forficula auricularia, common earwig, handsome little things. Macrolabic form in particular is beautiful with these long forceps at the back, and This is Uberelia anulipes, the ring-legged earwig, and it's got little dark rings on its legs. It has no wing cases whatsoever to show for itself, and quite fat, stumpy, broad pincers. Labia minor is something you could probably record across your county quite easily if you wish. Go look in compost heaps, but dung heaps. I look in dung heaps specifically for this, and it's often there. It's got golden hairs, it's quite short, quite small, slender pincers at the back. The wings do protrude and it does fly, but it's quite small. It's only up to about 6 millimeters long, and it's a lovely little thing. But you can search it out, but you've got to search out in, go and look in stables and farmyards and piles of manure at the side of the road, and you could easily just record hundreds of new sites for this across your county, and just put dots everywhere. So do that. So finally, no, not quite finally, but barklice. I love barklice, as they used to be referred to. I will call them barkflies. The sockets, the sockets, Pseudoscorpion. This is the new version of New. So Timothy New's book, that's the new version of it. You can also get the old version of New available online. Can be identified to an extent without much difficulty on external characters and wing venation, and there aren't many wing veins, so they're quite easy. I really am very fond of barkflies. Not a lot of dissection required and genitalia preps. Go on to the barkfly recording scheme website, and there's a key which is great. It's got lots of pictures, and it's step by step. Is this even a barkfly? It takes you through with pictures of the wing veins, and it's tremendous. I really like it. You just brush them off trees or twigs, in the most part. Smaller than 7 millimeters, generally brown, grey, yellow. Might have wings, might not. They don't jump. The psyllids jump. The plant lice and springtails obviously jump. They tend to run away rather than fly away, so they're quite easily brushed off a tree with a pasting brush or other. They have long thread-like antennae. They have a domed face and quite simple wings, and so and they're around now. So now's a really good time to try out the key online on the barklice recording scheme and try looking for barkflies. They are around some species all the time, but now's a very good time. So quickly, they have 2 triangular cells. That's how you know it's a barkfly. These triangular cells at the end here, which you can see, they have long filamentous antennae that separates them out from a few other things. But they have this bulbous nose. So this postclypeus, they're quite bulbous. And nice things, and say they don't fly away much when you find them. When you're looking at the key, the wing venation is quite straightforward. You might be looking at whether there are hairs on the veins, but also there's this thing called an areolepostica cell. It might not have that. This might be connected to the median vein, but that's it. And it will break down a lot of the key just in learning what that is. So that's the areolepostica key. So there we have one barkfly and some of its characteristics. So there's that bulbous postclypeus nose at the front, long filamentous antennae, and sometimes dark patches and patterns on the wing. It's not triangular there, but you can see it's still effectively a triangular cell. And you're looking at how dark these things are, what patterns they are, and quite where things join. So not many veins to worry about. The external genitalia characteristics can be helpful. You're not necessarily having to dissect a great deal. So some of this one you can recognize from the outside. So nice group to do. So give it a go. This is one found only the other day. It's lovely Ectosocus petersi. They were both sitting by these little egg masses on an elm leaf. There was ash leaf, and they cover their eggs in sort of debris and mess, and then a little tent of web over the top there. And there we go. So that's that. This is an Ectosochus briggsii, similar species, Graphosochus with really distinct dark markings. Mesosochus without any wings at all, but like a radiator grill for a nose. Interesting patterns. This is a barkfly that looks a lot like the springtail we just showed, except this bird carrier doesn't sit on tree trunks, it's in leaf litter. So you might see this running around, you might think it's a springtail in the way that I thought that springtail was a barkfly. Bird carrier, recognise ultimately when you've got one. It has a couple of dark lines on the labrum at the front. And to show that new discoveries can be made with these, this was new to Britain in 2015. I found this about 2 weeks ago, new to Derbyshire. It's basically yellow. It's got a dark line between the eyes and the antennal base there. The patterns on the wing are also distinctive. And this comes from Chile. So it's a Chilean barkfly, which is cropped up here, and it's reasonably easily identified, but new to Britain. So finally, this really is funny. So from sockids to cockids, these are things which are probably about as difficult as anything I could possibly have talked about this evening, but really interesting. Males don't have wings, females are, or males do have wings, females are always wingless and virtually don't move. Generally, quite often immobile, very flattened, possibly produce waxy structures to cover themselves in, very unlike insects or any kind of animal for that matter. They look more plant-like. They feed on plant saps, sucking at the juices of a plant, sometimes fungal juices. Can be a serious pest. The cochineal scale is well known for producing red dye, red colour stains that we use in microscopy, and also often in other things, other food dyes. So people with red lipstick on may well be coating their lips with dead scale insects. So these are the ones I particularly like, the ensign scales. Each one of these I found was new to the county because, let's face it, nobody sees them, nobody's looking for them, and no one can identify them. But ensign scales are not that difficult. Arctothesia cataphracta, lovely little thing. This wax that it's exuded in a very specific pattern. These three little scales of wax on the back there help you to recognize that species. And you know, when it turns over, you realize it's an insect. It's got legs and sucking mouth parts. I found this in sphagnum, in a sphagnum bog, digging among the sphagnum. Tiny little white flecks turned out to be the ensign scale insect. Likewise, the moss ensign, found it in damp moss. I think this was a New County record as well. Orthetiola vejovskii, and slightly different arrangement of these waxy patterns on the back, but lovely little things, really, really nice. And no one recognizes them as an insect. So now that you do, you can start recording them potentially. Neustedia flacosa, number of papers out there where this is new to the area. Slightly different arrangement of these scales. You can imagine these are waxy extrusions that grow out of the insect. Where it's backlit there, basically the insect is this bit in the middle, and these petals have grown out. Quite often when you're taking things like this seriously and you're studying them, you have to dissolve away the wax. It's helpful to identify some of these distinctive ensign scales, but in other cases you need to get down to the insect itself to identify them, which is where the complication lies. So there are all sorts of different cockids, and when they're not things that are covered in wax, they look like a little disc of plant material like this. Very hard to recognize as having legs of any sort. These are mealybugs, same family, which look a little bit more like an insect. This is a very flattened, This is the same beast, really, really flat. You wouldn't know it was not a piece of plant matter. But when they produce the wax and fluff and various fluffy scales that you see on some trees, at least they're flagging up that they're there. You might see these on apples, these sort of scales, which obviously look like a scale. You flip it over and you can see little sucking mouth part there, a little abdomen around the top there, and these stumpy, perhaps wings or remnants of limbs. So it is an insect, but it just looks like a little scale of something that's been produced by the plant. But good luck with them. They're not straightforward, but there are helpful things out there to help us with. Chris Malumphy's a very helpful guy, sent me information in the past. This page this paper has got a key to British ensign scales. This scale net has got scale insects of the world. Scale insects of California is quite daunting because there are an awful lot. There's 3 volumes online about that. But you'll see a lot about scale insects, very useful information. Again, Chris Malumphy has written, this is the scale insects of Lithuania, very helpful, includes British species and information that's of use to us here. One about scale insects across Europe, and the Irish one is possibly the potentially most useful, but again, more information with descriptions. So scale insects, finish with something really, really tricky. But there are other things you could look at. I'm not going to go into. I'm not even going near the aquatic stuff. I've gone on far too long. But in doing this, starting looking at these things, they are under-recorded. All of those things there were new county records. That was a new country record that I recently found. So there are lots of new discoveries you can make around every corner. It's just a matter of starting to look. I haven't mentioned any more about harvestmen, but in the last 2 years, 5 species new to Britain, all very distinctive. So if some of the stuff I've talked to you about has been too daunting, go back to the harvestmen, go back to the millipedes and the woodlice, and still lots of scope. Just a final thing, just to plug there, if you've never used the Biodiversity Heritage Library, it's a good place to find old reference papers if you're looking for slightly more obscure things that there aren't books about. You find them, as I say, the usual places: turning stones, looking under logs, under bark. Collect them by beating of branches as well, sweeping if you're not looking for thrips, pitfall trapping, tall growth funnel extraction, all of these things, brushing trees, sieving, things like that. Pick them up on a paintbrush, stalk, spill, forceps, take a bit of the substrate if necessary. Most of them can be dropped into alcohol. Certain things like the thrips, again, a bit awkward, easier if you've dropped them into glycerin and acetic acid, which I suspect you won't, but can still be dropped into alcohol perfectly well and still examined and identified. If you're doing DNA, if you've got the opportunity to do anything with that, then you want a purer ethanol. I'm not going to go into biological recording. You all know about biological recording, I'm sure, except to say that normally with iRecord and other things, you might well include a photograph in the record. With a lot of the things tonight, you probably need to keep a specimen to verify it later down the line if necessary, and certainly to identify it in the first place. So, and even though they're obscure, you can record them on iRecord. So this is that Chilean barkfly. So that's all right. That's on iRecord already. So good on iRecord. So there you go.
Thank you, Paul. That was really interesting. I learned a lot I didn't know. Yeah, I'm sure you've inspired lots of people to get out and look for some of these more obscure groups. So yeah, we'll go into questions now. So if anyone has a question, you can either type it into the chat, or you can give us a wave and let us know that you want to say a question out loud.
Okay, we've got a question from Tia. What is cocked wax used for?
It's just, as far as I'm aware, it's just a protective device. I'm not sure. I've not got into them sufficiently to know very much about their life histories. It's been just when I've had to identify large samples of things down to families and orders. So they're one of the families I just find fascinating to find and try and identify, but I'm not really sure. My assumption is if you're going to exude a wax coating, then it's got some kind of defensive mechanism, I would have thought. Certainly a lot of the cockids have got, shells sort of structures which are protective. They're like limpets. But the why they then declare themselves by having candy floss spilling out from underneath and flagging up that they're there, I'm not quite sure. So I don't really know beyond that.
Okay, thank you. Lisa, do you want to unmute yourself?
Yeah, can you hear me?
Yeah.
Yeah, at the risk of hijacking you, I just wanted to say that the Queckit Microscopical Club has actually got a project on for Pseudoscorpion recording, and we would be very interested to hear if there's anyone that's listening in that would be interested in getting involved. We would love to hear from you, because the more people that we can get recording, the better. So if anyone's interested, they can either go onto the Queckit website and find out about it, or they can email me at pseudoscorpions@queckit.org. And we'd be really interested to get some more people up and down the country to improve what we're getting, because they're quite elusive little buggers.
Yeah, they are. I think I was speaking to Les at the beginning about the project, and yeah, I mean, they are great if you can find them. And obviously, if you're looking down microscopes, they're a great thing to look down the microscope at. They're marvelous beasts. Yeah, I remember just a silly little anecdote, but some years ago, we were filming harvestmen with the BBC for Autumn Watch. And while they were getting on with that, I was turning over stones, and I found some Pseudoscorpions. I found 4 under a stone. I said, well, you've got your macro lenses, film these, because you won't see any again. And they said, oh, no, we're doing harvestmen. As the BBC very focused, we're doing harvestmen today. So that's what they did, and they didn't film them. 2 weeks later, we got a sort of message around through people who do Pseudoscorpions saying the BBC are doing a program about back garden wildlife, and they're really keen to get some footage of Pseudoscorpions. Can anybody find us any? Of course, no. No, you missed your chance. But I think fortunately Liam, had some in his fridge trotting around, as you do. But yes, there was the opportunity missed. But no, you don't see them.
One of our members seems to have a bit of a magic touch, because I think he's had sort of 20 odd out of one sample. Nice. Which is something that the rest of us can only dream about having. Yeah. But yeah, we'll keep on trying.
To be honest, compost heaps, dung heaps are very good for 2 or 3 species, and just sieve leaf litter. What the secret is, get some leaf litter, sieve it onto a white sheet, and wait till everything else is gone, because the last thing to move will be the Pseudoscorpions, and then you'll see them, and they do. Then they'll rush, into a white tray, and they will start to move. And then once you see them, once you've found your thing, before you lose it, make a little ring in the leaf litter or the dust around it, so that you know where it is.
They're usually very easy to see anyway, because the palps are bright orange, and they're waving around all the time.
Yeah, I mean, some species are a bit more difficult to see than others.
Yeah.
Yeah, I've not had any luck with the white tray method. I've only found mine floating in alcohol with the funnel.
Yeah.
So far.
Yeah, okay. Yeah. It works well in a funnel. What was I going to say? Underbark, an ancient tree bark is nice for those genetted ones as well. The common churneys found in the New Forest underbark, but then they're not easy to find. They are very small and they don't move much. But when you sieve them, that spurs them into action sometimes. But I'd go compost heap. Urban farms that we've got in Sheffield, their compost heaps are great. But you've got to get mucky. You know, go to a dung heap. You'll find your Pseudoscorpions and your lesser earwigs, and a couple of fairly unusual woodlice. So get out into the piles of dung.
Thanks.
Thank you, Lisa. Yeah, if you want to put a link to your project in the chat, and then we can send out a link in the follow-up email as well.
It would be great to get more records. The other thing that springs to mind in terms of Pseudoscorpions is the bottom of marram grass. There's a species, the name of which escapes me, Latreille, but marram grass is quite good for some as well.
Okay, we've got a question from Julie who says, if I find something and manage to get an expert to ID it on Facebook, should I record it even if it is a common species? Absolutely.
I mean, first two parts, I'd answer that twice. I'd say yes, if an expert's identified it, they'll usually say, yeah, that's fine, you know, I record that. On Facebook, often people say, thank you very much, shall I record it? And they'll go, yeah, please do, or they'll ask you to do it. So yes, if an expert's seen it, absolutely. The other part, that my other answer is anything is worth recording. Record small tortoiseshell butterflies, you know, record everything. And especially if you can just get your eye in on one of those things I've talked about tonight, and you just know that thing, even if it's like an ensign scale insect, which you've recognized, just go looking for that and record it everywhere. You know, it becomes your thing, and you'll put docs and maps which will revolutionize the data. But yeah, anything common, always. And even when you're looking for these difficult things, record the common stuff that's sat next to them as well, please. You know, that everybody out there wants the beetle and the spider and the everything else records. So yes, please just record.
Okay, we've got a question from Andy or Jean Young. Is the oxeye daisy red larvae the only red thrips? I had one the other day and thought it looked pretty distinctive.
I want to tell you the answer to that because I want to know. I don't know. I got it's on the front cover of the Kirk book, and I've read it the wrong way round, in that I've read about the oxeye daisy and the species that are on oxeye daisies, and that the oxeye daisy leucanthemi thrips, whatever it is, leucanthemi has this distinctive red larva. What I haven't read is, I've not read about everything else. My knowledge of there are a lot of thrips, and I'm not sure. The disadvantage of that excellent thrips key, the lucid key I mentioned, it's really good, but all of the pictures are of microscope specimens, and they're all pretty much transparent, and it's features of legs and heads and bodies, and very few whole animals and live animals. And so I'm not sure, and I've started to go through it. I was going through and reading the descriptions to see if there were certain species which are absolutely unique, that will only appear on, say, ragwort or something like that. so that I'm really pretty sure of what it is. And no, I wanted to find out. So I think, is it always that red one there? And I suspect it isn't. I suspect there's another red one, because I've seen it before in a sweep sample, and I'm not convinced that there was oxeye daisy where I was sweeping. So that's as much as I can say. I've seen it before, and I don't think there was oxeye daisy there. So I don't know. So as I said at the beginning, you can ask me all these questions, but I can't have answers for you. But I'd like to know that if it's the only red one, but I don't think it is. But if you found it on oxeye daisy, I'd be pretty confident that that's the one. So yeah, so if there's a red larva on an oxeye daisy, I'd go with that.
Thank you. Don't think we've got any more questions. Does anybody want to ask anything before we finish? No? Oh, Julie, you got your hand up?
Yeah, just on the subject of where you find the insects, is it useful when you do the eye record to say what type of plant you found it on? If it was on a specific kind of plant and you know what kind of plant it was on?
Yeah. Definitely in those things where we know that it's a helpful thing, like the thrips. Any kind of, the more detailed, I mean, we're all a bit lazy about it. I mean, I'm as guilty as anyone in that I'll probably say, you know, I found at this site. I might not even say I found something on dead wood. I might not even say, you know, I certainly wouldn't go to the details of what sort of dead wood, but it would be helpful if I did. And certainly when I'm entering a record and I record on a computer, I will add a lot more information than I tend to do when I'm recording it on the app in the field. Although there is a comments part of the app when I'm recording, I'm not in a position to sit down and add I've not got the time. I'm usually trying to record. But if I'm entering in details, you know, just add as much as you can. Certainly if it's a thrips, what plant it was on, without a doubt, include that information. As with weevils and various beetles and, you know, gall wasps and things like that, you know, it's a really important piece of information to, and actually I'm becoming a much better botanist from looking at thrips and some of these things. I'm an appalling botanist. But I'm learning plants because I've always learned plants because of looking at weevils and things and needing to know. But I'm not interested in the plant. I'm interested in the weevil or the thrips or whatever. So yes, definitely 100% record as much information, but certainly if you know the species of plant, that'd be great.
Several years ago, I got a white buddleia in my garden, and I was interested in a small insect that was dipping itself in and out of the florets, and I made a small video of it and sent it off to the Quacket, and the forum came up. Yes, it's undoubtedly a thrips,
right?
But I couldn't have recognised it. But
No.
it's very interesting. It spent a long time, and it was wiping it's sort of stroking its wings, presumably to get rid of some of the pollen.
Yeah, they're fairly fastidious. They're a really interesting thing to look at. I mean, they're really small, so you need a very good sort of, it's the sort of thing where a magnifying glass is slightly better than a hand lens. It's slightly more convenient to sit staring through a magnifying glass. But you know, when you read, say this book by Kirk is really fascinating about the battles that the males have and the sort of courtship and fights that are going on, and they sort of fight to the death. It's fascinating stuff. Thrips could be the new, I'm going to say, I almost said butterfly, then definitely not going to be the new butterfly. They're edging into, you know, they could become popular. But the problem is preserving them and preparing them on a microscope slide. It goes beyond something you can do on the dinner table, really, in between eating your sandwiches or whatever. So you kind of need to be set up to do something like thrips, because you may have to clear them in all sorts of things like acetic acid, but I use clove oil. But yeah, so you've got to clear them and take them through stages of alcohol and use complicated mounting materials, which is why I read somewhere recently about using glass glue. And so I'm going to give that a go, because you just drop it onto your slide and then it bonds to the slide and the cover slip and is very, very clear. So I don't know if it'll work very well, but I'm told it does. But yeah, thrips are a nuisance in that respect. Nobody said this lot were going to be easy. There we go.
Okay, so yeah, I think we'll finish there. The next webinar is on crane flies with Pete Boardman on the 9th of September, which is 2 weeks today. So do join us if you can, and thank you and enjoy the rest of your evening, everyone.